Our next speaker is Dr. Sara Federico. She's a member of the St. Jude faculty and director of the Solid Tumor Division. She received her bachelor's degree at Stanford University in California and her MD at Virginia Commonwealth University Medical College. She did her internship and residency at Lucille Packard Children's Hospital at Stanford and her fellowship at St. Jude. She is author and co-author of numerous publications and articles, and her research interests include clinical investigation of pediatric soft tissue and sarcomas, translational research, pediatric solid tumors, and novel drug development for pediatric solid tumors. She will be talking today on the current trial updates at St. Jude. Dr. Federico.
Great. Well, hopefully I don't put you to sleep since you just ate. I'm going to stand to the side just because I'm not tall enough to see over the podium. So I really appreciate the opportunity to be here today. Neuroblastoma is like my real passion, and we were just chatting about why I went into being a clinical trialist, and it's really because I want to see those survival curves, especially for the patients who have high-risk disease, increase. So I have no financial disclosures. So, I hope that with today's talk and through all the different folks that you've been listening to, I hope you get a sense of the collaboration. So there really is a collaborative spirit in the researchers who take care of patients with neuroblastoma and who are driving research for neuroblastoma. And the truth is we need each other. We need to be asking similar questions and trying to answer them in different ways so we can come up with new therapies and make changes. And so the curve on the left, or the line on the left, really just highlights some of the changes that the cooperative groups have made over time, focusing on high-risk neuroblastoma. On the right, kind of the standard of care in the US and in Europe is really to give therapy in 3 different sections. I'm talking high-risk therapy, induction, using multiple different chemotherapy agents, surgical resection of their primary tumor, consolidation being high-dose chemotherapy with stem cell rescue and radiation therapy. And then the biggest change truly in terms of improving outcome and survival came with the addition of an anti-GD2 monoclonal antibody at the end of therapy. And so over time, there has been a lot of progress, incremental progress for patients with neuroblastoma. So to orient you, on the left hand of the slide, those are survival curves. So on the bottom, it's time, and on the y-axis, it's overall survival. And you can see that the top lines, the green and the red line, are really the long-term survival data for patients who have lower intermediate risk neuroblastoma. So they've really come up, and actually, over time, what many groups have been doing is starting to pull back on the amount of therapy with the hopes of limiting toxicity for those patients. And then on the bottom, that's the curve for high-risk neuroblastoma. On the right-hand side, you can see that is the Kaplan-Meier curve for high-risk neuroblastoma, kind of broken down by time periods. And so to orient you, on the bottom in black, that was like the 90s. And then you can just see that over time, we have made progress. And by we, I mean the neuroblastoma community has made progress, but it's still not 100%. And still, this therapy comes with an incredible cost and toxicity. So we have a lot of work to do, but there has been progress. And so today, a lot of the stuff that I'm going to talk about is just building off of the antibody work. So this is the first kind of the seminal paper of Alice Yu, when she presented the data on AMBO-032, which was basically adding the antibody to the end of therapy in the post-consolidation setting. And patients who were treated on that trial, they all received induction chemotherapy with surgical resection, transplant, and radiation. Then half the patients were randomized to receive immunotherapy, anti-GD2 antibody therapy, and isotretinoin, and the other half just received isotretinoin alone. And in interim analysis, when you pause a trial and look to say, like, are we seeing too much toxicity, or are we seeing exceptional or not poor responses, they actually saw this 20% improvement in the patients that were receiving the antibody therapy at the end of treatment 20% more of them were surviving. So that's when the trial stopped, and that's when antibody therapy became standard of care and got added to the end of therapy for patients who have high-risk neuroblastoma. But again, we still have a long way to go. And even though you can see on this curve, it says that the 2-year event-free survival, meaning if you haven't had an event, it means you haven't relapsed or passed away from a different reason, is 66%. But you have to remember that these are patients, that doesn't take into account all the patients. And so these are patients who had done really well in induction, really well in consolidation, then enrolled on this study. So actually, when you look at the most recently completed COG trial called AMBL 532, the 3-year event-free survival, so again, event-free survival meaning that you haven't relapsed or passed away from another reason, including all the patients, is only about 51%. So there are a lot of ways that we can target neuroblastoma. I like this schematic that Dr. Granger put together with Dr. Du Bois, but we're just gonna focus on antibody therapy today and some of the work that we're doing. So again, like, this is terrible, but it's how I always describe it. So tumor cell antibody, it's like a flag that hangs off of the, antigen GD2, it's like a flag that hangs off the tumor cell. When we give antibody in the central line, it's going and it's looking for the flag, and it binds to it. That doesn't kill the tumor cell itself, but then your immune system can now recognize. It didn't recognize tumor alone, otherwise it would have killed it. But you put an antibody on it, and you rev up the immune system, and now all of a sudden the patient's immune system says, oh, I see you, and I'm gonna destroy you. And so that's what the therapy, that's really the mechanism of action, how it works. So at St. Jude, we've had the opportunity to work with a humanized antibody, which is a little bit different. So the antibody that was used in the first initial studies is called a chimeric antibody, which means it's part mouse, part human. This antibody called HU1418K322A, which I'll just call HU1418K, it's mostly a humanized antibody, so about 98% humanized. And you can imagine that if something goes in your body that's more human versus something that's part mouse, you might have less reactions to it, and so there are less reactions, so it's less immunogenic. It does have a single point mutation, which is highlighted there in the green, which basically helps with alleviate some of the symptoms, and specifically some of the kind of leakiness when patients can get puffy and fluid can go to the wrong places, or some of the pain symptoms. And because it's a humanized antibody and it's better tolerated, we're able to give a higher dose. And so the dose of the antibody that we use is about 2.5 times the dose of the dinutuximab that's used in the other studies. I should say that nobody knows, like, what is the best antibody. Like, no one's ever done that study, because people ask that all the time. I don't know. I wish we knew, and then we were just using that, but we just don't know that data. And so when we had this antibody, we've done, at St. Jude, 3 trials to date. So the first trial was a phase 1 study. So again, phase 1 study, just looking to see what dose can we get to and give in a safe manner, with the hopes being that it's going to decrease the tumor burden. And so in a phase 1 study, there were 37 patients who enrolled, and there were some patients who developed response. And so these are in patients who had relapsed disease, and we determined what the dose was. The subsequent study, the next study, was really to say, well, what can we combine it with? And again, these are like 2010 era. Okay, so right after kind of Alice used that first dinutuximab study came out. So the next study was to say, well, geez, can we combine it with chemotherapy? And now it seems like that's kind of a no-brainer, because that's what a lot of trials do. They combine chemotherapy with antibody therapy. Back then, everyone thought we were crazy, because chemotherapy suppresses your immune system, right? It knocks your immune system down. And we just described that you have a tumor, you have antibody that binds to it, and now you need a immune system that can go fight it. And so everyone in the beginning said, this is kind of a waste of time. Like, what are you thinking of doing? You're going to give chemotherapy, knock it down, and chemotherapy and antibody aren't going to work. But there's enough kind of data and literature in the adult space in different cancers and in adults who had bulky disease that suggested that maybe actually when you give an antibody with chemotherapy that you can still get a response. And so that's why we went forward with this chemoimmunotherapy study. So there were 13 patients. They all had relapsed disease who enrolled on this study. They got different chemotherapy regimens in the first 2 cycles, the next 2 cycles, and the next 2 cycles, because we were really trying to determine, is it safe to give the antibody with chemotherapy? And we were hoping that it would be safe and that it would work. And what we found is that we saw responses. And so kids whose cancer had come back and they had relapsed started having their cancer go away. Now, the trouble with that study is that it was only 6 cycles long, and that's just not enough to sustain. Most patients need much more therapy than that. We did note that there was this kind of delay, even though many of them, they came off after 6 cycles. There was this much longer time period from finishing therapy to when their cancer returned in comparison to other prior chemo studies. And around the time that we were publishing that data, then the Children's Oncology Group was publishing their data of AMBL 1221, which was a phase 2 study that basically had 2 treatment arms. So one treatment arm got irinotecan, temozolomide, and temsirolimus, so 3 chemotherapies. The other treatment arm got irinotecan, temozolomide, and dinutuximab. And at an early time point, again, these trials always have built-in mechanisms to make sure that we're not causing too much toxicity and to look for early signals one way or the other. At an early time point, at the interim analysis, they actually saw that the patients who received the 3 chemotherapies, irinotecan, temozolomide, and temsirolimus, that only 6% of them were having a response, meaning their cancer was getting smaller. And this was in stark comparison to the patients who were getting chemoimmunotherapy, so irinotecan, temozolomide, and dinutuximab, where about 53% of them were having a response. And so the trial was halted. No more patients went on to the 3 chemotherapy arms. Instead, more patients were added to the chemoimmunotherapy arm to get a better sense of what is that outcome and what sort of response are we seeing. And so on the right hand of the slide, then that's the follow-up, where patients, they expanded the cohort of patients who got chemoimmunotherapy and found that actually when you added in those patients, that it ended up being about 40% of those patients had a response, meaning that you could see visible regression of their tumor. Another 40% had stable disease, and it's complicated how we grade response in neuroblastoma, but that just means it wasn't growing, so at least that was a benefit. And honestly, some of the reasons some of the patients had stable disease, many of their metastatic disease sites went away, but if they still had a big tumor in their abdomen, that one didn't really change in size, so they had stable disease. And then a smaller percentage of patients didn't respond. So when we saw our data and we saw the COG data, then we said, well, we got to take this up front in newly diagnosed patients. And so that's this trial right here, MB 2012. So what MB 2012 was it was a single institution, meaning it was just only done at St. Jude, a clinical trial, phase 2 study, looking at chemoimmunotherapy during induction. So to orient you, for induction, those are the circles and the squares on the right. Patients received 6 cycles of induction. That was kind of the COG standard around that time, although now it's really more like 5. We added antibody into each induction cycle, so starting with induction cycle 1, and then there were these different time points at which we would measure response. We knew when we added antibody therapy that it comes with side effects, and that if we were going to add antibody therapy with chemotherapy, that we really wanted to see a big improvement. And so when the trial was first designed, we looked after 2 cycles of therapy, and we knew that in patients treated on prior COG studies, when they looked at that 2-cycle time point on AMBO2P1 and then actually later on a larger trial, that when you took pictures after 2 cycles, 40% of patients had a response to therapy, meaning some criteria that where their tumors were getting better. It doesn't mean the others weren't getting better. It just means they met the criteria for a partial response. And so what we said in this trial is, if we're going to add antibody, and we know that's doing a lot to our patients, we want to see 20% improvement. So we set a bar that said after 2 cycles of chemoimmunotherapy, we want to see a response rate of 60%. in order to say, okay, we should study this even further. Induction is followed by transplant. In this case, for this clinical trial, we used a slightly different chemo regimen for transplant, and that's because at the time that we wrote this clinical study, SioPen had just conducted a trial where they looked at 2 different treatment regimens for transplant. In one of the regimens, the patients got busulfan and melphalan, BuMel, and the other regimen, they got SEM, carboplatin, etoposide, and melphalan. And in the SioPen data, BuMel was better, which is why we went with BuMel and just a single transplant, followed by radiation. And then at the end of therapy, patients got immunotherapy, but using the humanized antibody. And again, also at a higher dose just because that's what we could give. And so after the first 42 patients, again, we paused and we looked and we saw that actually the response rate after 2 cycles was better than what we had expected, or that we set the bar at. It was actually 76%. And it fit because we were giving chemoimmunotherapy, and you could just tell kids were getting better a lot faster than they had previously. And so what we did at that point in time is we added more patients to the study, where we would then be able to get more robust numbers to see if this is really true. Is this just luck, or is this statistically sound? And so that's what we did. Added more patients. And then this is some of the data. And so what we found, to orient you, in the top box, this is a waterfall plot, which is showing the response of your primary tumor, so the one that started it, and you want to go down. And so the one all the way to 100 means the primary tumor is completely gone. The color of the bars is actually showing their response at the different time points. And then there are these letters based on if they had bone marrow disease or not. But the bottom line is, I think when you see the response to the induction therapy, so 6 cycles of chemoimmunotherapy, we saw this dramatic response where 96.7% of patients had greater than or equal to a partial response. And so to give you an idea, historically and this is looking back over many, many trials through SioPen and actually through the Children's Oncology Group, this number of PR or better is usually around 74 to 80 maybe. And so we were seeing this early response. And their bone marrow, none of them had more bone marrow disease. It all cleared very, very quickly. And none of the patients developed progressive disease, worsening disease, during induction, which historically that's about 7 to 10%, depending on the trial. And so, again, we were very excited. We were very hopeful that that would translate to long-term survival. And indeed, the 3-year event-free survival actually ended up being 73.7%, and the 3-year overall survival of 86%. And so again, just to orient you, the AMBO 532 3-year event-free survival for the whole cohort was about 51%. And so a big improvement, suggesting a signal of activity. But I say that, though, recognizing that in any time people present data, it's like my pet peeve when people get up and act like used car salespeople or whatever, where they're like, it's too good to be true. Like, we have to present the limitations, right? And so this was only at a single institution, right? And so it's a good signal of activity, but it was only done at one place, and it really needs to be done in a randomized fashion with a whole large group of patients, where you're gonna get a bunch of different biologic differences between patients, and see if it's really true before it becomes the standard of care. So again, I was very excited, and my patients are doing great, but that's just why it needs to be validated in a larger randomized study. In the last session down at ANR, we actually just presented the 5-year outcome. And so one of the thoughts has always been, well, are there going to be patients who develop later relapses? Because typically for patients who relapse following high-risk neuroblastoma therapy, if you're going to relapse, about 80% will relapse in the first 2 years, and then that number drops off significantly. So we just didn't know if we were delaying relapse, and we aren't. And so the encouraging and exciting thing is that the 5-year outcome really looks the same. So 5-year event-free survival was 70.2%, and the 5-year overall survival of 85.8%. It doesn't go forward anymore. Oh, there we go. Now it does. Okay. And so what does that look like when we compare it to historical controls? And so just to orient you, on the left hand of the slide in the blue bars, so in the red bar box on the bottom, that's 3-year event-free survival, and then comparing it across clinical trials. So the blue bars are the most recently completed COG trials, which the 3-year event-free survival was 51 and 55.6%. The purple bars, is the 3-year event-free survivals of the most recently completed Siopin trials in Europe, which is 34 and 44%. And then the green bar is the St. Jude trial, which was 73.7%. Again, but single institution study. So at the time, it really bugged me because I was seeing all these encouraging results, but then we were just only doing it for kids that were treated at St. Jude. And so at that point in time, talked with the Children's Oncology Group and said, okay, we need to try this outside. Again, these are early days. Now I know people were used to giving chemoimmunotherapy, but there was a lot of concern early on that people wouldn't know how to take care of these patients. So we conducted a COG study, a pilot study, to basically say, is it feasible to give at different institutions safely? And so that's what this was. So AMBL 17P1 opened at 10 centers. There were 9 centers that actually enrolled patients, both inside and outside this country, 42 patients with high-risk neuroblastoma, and patients got dinutuximab in induction cycles 3, 4, and 5. Now that's a big difference, right? Because in the MB 2012 trial, they got antibody through all cycles. In this study, they only got it in cycles 3, 4, and 5. And the rationale for that was that, sometimes when patients initially present with neuroblastoma, they have such significant disease everywhere. They have big pleural effusions and a really big belly and be very, very sick. And if you give antibody and they get a little bit leaky, then you can cause something catastrophic. And none of us wanted to do that. And again, these were early days. So that's why the decision was made not to give it in the first 2 cycles, because we didn't want to hurt patients. And then there was a lot of question, although I never agreed with it, but there's a lot of question about whether or not you would be able to collect stem cells okay. And so that's why it was done after stem cells, which is not a thing. You can collect stem cells just fine. And so at the end, what we were able to say is that these patients were able to get dinutuximab with induction chemotherapy just fine. And now we're starting to look at that longer-term data, and hopefully should have that coming together later this year. So my question then is, how can we build upon this? And we need to do the randomized study. And so that's what this is. So AMBL 2131 is a current phase 3 chemoimmunotherapy study being conducted through the Children's Oncology Group. That's the primary study team together. So myself and Thomas Cash co-chair this. And then our vice chairs, and so Thomas Cash is at CHOA in Atlanta. And then the vice chairs are Naveen Pinto and Carrie Strebe. Naveen is in Colorado and Carrie's in Ohio. And so what this trial is really doing is it's asking the question, when we give immunotherapy, anti-GD2 therapy, to patients during induction, and we do it in a randomized way, so patients all get the same first cycle of chemotherapy, and then they're randomly assigned to receive either chemoimmunotherapy or chemotherapy alone, followed by all the rest of standard COG therapy. Do we see an improvement in outcome? So I don't know the answer to this question because it's ongoing. This trial is ongoing. Importantly, we wanted to make sure, so when we were designing the study, I said, well, I feel conflicted because even though it's single institution chemoimmunotherapy helping patients, and we need to ask the question in a randomized way, is there equipoise between the 2 treatment arms? And so what we included in this study is to make sure that if patients who were receiving standard therapy were not having a good response, that they could get extended induction, which is in the red box, with chemoimmunotherapy on study, with the hopes being that they would then have a response and loop back into the regular study. Extended induction, I don't know why it keeps getting stuck, is just chemoimmunotherapy, which is what that first trial was about. So irinotecan, temozolomide, and dinutuximab. And the patients can get up to 6 cycles, so they do 2 cycles. We do a disease evaluation, make sure we're moving in the right direction. and then we can give up to 6 cycles before going on to consolidation. So that study activated in the US on tax day of last year. There are about 160 sites now that are up and running, and actually this week it's kind of closer to probably 150 patients have enrolled. So again, it's early days. We don't have the data. It should take about 3 years total to be able to accrue 200 patients per treatment arm. And then, of course, we'll have to wait because the marker is 3-year event-free survival. So we're not going to know the answer to the question for about 5 years, which I know feels like too long. It's just the way that it is, because we need to be able to see if it's statistically significant. So while that's going on, though, we need to keep pushing the field. And so what I've been super passionate about, and many others are also passionate about it, is how can we build upon the chemoimmunotherapy backbone? Like, how can we make that therapy better? And so one of my colleagues who used to be at St. Jude, but now is at the NIH, did a lot of this work when she was doing her postdoc, Dr. Wynn. And what she found is that IL-15, which is a cytokine, it really enhances natural killer cell cytotoxicity and ADCC. So again, tumor cell flag, GD2, is not on most cells in your body, which makes it a good target. We give antibody, and then the immune cells, the cells that actually are the ones that do the work and get rid of the tumor cells, are called NK cells, so natural killer cells. So this question is really saying, like, what can you do to natural killer cells to, like, rev them up, right? Like, let's get them going. One of the things we do is GM-CSF, which kind of can help increase the production. But another thing is, can we give a product, a cytokine, that will help increase production and increase kind of how those cells operate. And so when Dr. Wynn did this work in the lab, she basically showed that if you look at, this is in healthy donor cells, so no one that has neuroblastoma, but if you took if you took participants that were willing to donate their NK cells, and then you put them in a dish and you just treated them. So you just left them alone, those are the resting ones. The next dish you put IL-2 on, which also increases NK cells. And then the final dish, you put IL-15 on. And then you just looked at the activity. Actually, when you put IL-15 on them, the bars go way up because that really makes them lyse the cells. So the higher the bar means the more lysis and killing of the tumor cells. So that seemed like a good idea. And then she did some nice work in a mouse model where these different mice who had neuroblastoma, she gave them different therapies. So group 1 just got irinotecan and temozolomide. Group 2 got irinotecan, temozolomide, and GM-CSF and antibody. Group 3 got irinotecan, temozolomide, and IL-2 and GM-CSF. And then group 4 is the one that got chemoimmunotherapy plus IL-15. And so here, the bars are kind of the reverse. So growing tumors, the bars go up. Regressing tumors or killed tumors, the bars go down. And you can see that clearly group 4, the group that got the IL-15, did a lot better. So based on that work, I have spent a lot of time trying to put together a different clinical trial, which is using an IL-15 product. So currently, there are a couple of different IL-15 products that are available. One of them is very short, has a very, very, very short half-life, and there's a trial that should be coming, potentially using that with chemotherapy and antibody and IL-15 that has a different half-life through the NIH later. But what I wanted to use is a different product. So there's a product called N803. It's also previously called Alt803. And what it is it's an IL-15 superagonist. And so it essentially sticks around in the body a lot longer, does a really good job at, like, expanding and activating your natural killer cells. So there was a trial this product is owned by a company called Immunity Bio now. I mean, it's like changed hands many times, but that's who owns it now. And there have been some studies that have been conducted in adults. So this is a non-Hodgkin lymphoma adult clinical trial in which patients received this IL-15 superagonist, and they were able to demonstrate that it does activate and increase the number of NK cells, as well as CD8 T cells. Importantly, it does not increase a different type of T cell, which IL-2 can actually increase a certain type of CD4 cells. And so it does not do that, so that's really important. And in this patient population, non-Hodgkin's lymphoma, when they gave the treatment, it worked for a bunch of patients. The one thing that you can see here is that it does cause some skin irritation. It's an injection that's given subcutaneous. You can actually give it in the central line, or intravenously and subcutaneously. When you give it in a central line, there can be a lot more toxicity, and so that's not the preferred route. So giving it subcutaneously is the better way to give it. The results in terms of side effects is that you can get a bunch of NK cells that go to where the injection was and get some redness of the skin. But it's important to note that this is not an infection and it's not painful. It just looks red. But they, on the adult study, biopsied it, and there's nothing bad that happens with the skin. But I think it's important to know that. And so, again, when they gave this IL-15 superagonist with another drug called rituximab in non-Hodgkin's lymphoma, they got a bunch of these responses. So down means their tumors went away. And then there's a swimmer's plot next to it in C, which shows green are complete responses. That's what CRs are. And a lot of these were patients who had previously been treated with rituximab and failed therapy. They got rituximab again with the IL-15 superagonist and responded. And so I have spent a long time, and it's going to happen, it's going to happen, trying to get this study open. So it's called Honeybee 803. It is a clinical trial, which is basically looking at the addition of this IL-15 superagonist. It's taken us too long to get it open because it's hard. Dealing with companies is sometimes difficult, but we are getting there. We just need one more contract signed. And so what this clinical trial will entail is basically it's the first time that IL-15 has been used in pediatric patients. It has been used in adult patients. And so the first group of patients we have to be a little bit more cautious with just to make sure that it is tolerated. I fully expect that there's going to be redness at the skin area, but we need to make sure that we don't see anything else that is unexpected. And what these patients will get is they'll get irinotecan, temozolomide, and then they'll get that humanized antibody. So the one that we used in NB 2012, where we give a higher dose, that actually has been manufactured to use in this study. So it's a little bit different than the dinutuximab from other clinical trials. And then they'll get the injection of the N803. As long as the first small group of patients is able to receive that safely, then it will go actually to a phase 2 trial where patients will be randomized to receive either irinotecan, temozolomide, humanized antibody, or irinotecan, temozolomide, humanized antibody, and the IL-15 superagonist. And then we'll look and see which treatment arm does better. Patients who have previously received irinotecan, temozolomide, and antibody will be allowed to enroll on this clinical trial as long as they didn't develop progressive disease while they were on it. And actually, there was a whole session right before this one that was talking all about these different clinical trials that have looked at patients who have received chemoimmunotherapy multiple times using different tweaks in their regimens. There's a whole group of them, usually about 50%, that will keep re-responding. And so even if they've received it previously. So this study should open, let's hope at the end of the summer. So everything is ready to go. The FDA is approved. We made the antibody. All the orders are ready. We have the date. Everything is ready to go. We just need one signature on a contract. And it will happen. It will happen. I think people finally are giving up on me. They're just like, oh my gosh, make her stop talking. It will happen. So hopefully, again, end of the year. It will open at St. Jude initially, and then the goal is in partnership with NANT, which is, I think you guys heard from Araz Marcellian yesterday, talk about NANT and the consortium and some of the clinical trials that are available. it will open at a bunch of the different NANT centers. It's not a NANT study, and that's just because we manufactured the antibody for this trial, and so it has to stay a St. Jude study, but it will be open at multiple sites. So hopefully it will be easier, and more people will have access at different centers. So I am immensely excited about this clinical trial, and can't wait to see if it really helps that backbone work better. And so with that, it takes a village. I feel incredibly blessed that I work with a bunch of just truly amazing people. For clinical trials to happen, of course, we need people to take care of them. Megan Delaney is the one in the circle with the blonde hair. She is our head APP at St. Jude for our solid tumor team, and our clinical team is really unbelievable. There are a bunch of other people that are up on the screen as well. With clinical trials, you need a big clinical trial team, people to work with regulatory and to partner with pharma. I'm appreciative of partnership with ImmunoBio. And then there are some different funders that are there as well. And then, of course, this is what keeps me up at night. It's who I think about every morning that I wake up. These two little ones. When I started my fellowship, I knew I wanted to do clinical research, and I really knew that I wanted to probably do clinical research taking care of different solid tumors, because many of the solid tumors, just the cure rate is not as high as it has been. And then in that first year of fellowship, I took care of the little fireman and the tiny red dancer, and they have become my passion and my reason to make sure that we move this field forward, so more kids can live. So with that, I'll take questions. Thank you.
Thank you, Dr. Federico. We have a question?
Hi. Thank you so much for presenting that, and it's very exciting, the new study that hopefully you'll have online pretty soon. I guess I have a question, and I feel like you're a good person to address it to, just from the sense that I got you talking about, you're at St. Jude, you're doing a lot of immediately applicable research. You go into clinical studies. I know that having spoken to a lot of patients, our main goal is to cure our own children. And so it does become frustrating when we have to wait for a long time. You see your own child decline, and there are these options, but they're not available to you. And so when you see something like the results that you get from the chemoimmunotherapy up front, that really is striking, just knowing what the other outcomes have been. And I know that randomized trials are the gold standard, but I also feel like more and more as we come here, where you see different doctors talk, people are able to find ways to get these types of treatments that are, clearly very successful to the patients more quickly. So I'm thinking about, for instance, DFMO, they were able to do that. I don't know exactly what the statistics were, but kind of the retrospective study. We know people who have been on DFMO who've been very successfully, responded to that. What do you think are some ways that going forward, the medical community can find statistically reasonable and, kind of substantive ways to create situations where we don't have to wait for a large clinical trial in a rare disease where children, can't wait 6 weeks for treatment because the disease will progress so much? Are there things that the community can do to still, be cautious, not harm people, make sure that it's something that's been proven, but not necessarily have to wait for hundreds of kids to accrue?
Yeah, so super important question. And I live your frustration with you, but not on the same level. I never want to pretend like I know what it's like to be in your shoes. So I think there are a couple of things. Number one, what can the community do? I think we all can keep advocating. And then I actually think parents advocating helps. Maybe not in the moment, right? Because what you're asking for is an answer in 6 weeks, is your exact question. But I am seeing the results of parents advocating. So let me just give you an example, and of the community working to push things. We are limited by certain things. So one of the reasons we have to have scientific rigor is that the goal is to try and get approval for these drugs. Because then if you can get approval for drugs, you can also make it accessible to patients everywhere. And so in doing that, obviously you have to go through the regulatory processes and partner with the FDA. The FDA, most recently, with a company who's going to be helping the high-risk neuroblastoma patient population, in the past, they have really, really, really frowned upon doing combination studies where you're combining multiple drugs or agents together from the get-go, and they always say, we want to see single agents, so the one drug, and demonstrate that that's effective before we'll ever look at all this other data and approve it. That is just not reasonable. And the medical community, the research community has been pushing back against that for a very long time because, to your point, it wastes time and it just delays everything. And I have to say, I was on a call with the FDA 2 weeks ago in this company, and they are letting, I don't want to jinx anything, but they're kind of have abandoned that. So they are actually getting on board with the combo therapy. But it's because we've all been going and bringing it up to them. It's because families have been advocating. On that call, Nick Bird, who I'm sure many of you know and is amazing, he can bring a voice that when he speaks, everyone really listens to, right? And he can kind of call hey, that's baloney, right? I appreciate what you just said, but that's not okay. And so that is a way that we can partner, but it is frustrating. The other thing that's frustrating is sometimes just having access. So some of the studies, like the trial I just presented, I've been trying to get that study open forever. For way too long. And it's because I just can't get the drug. If you can't access it, and it's because sometimes there are ways to try and get around things. If a drug has been commercially, or if it's approved and it's commercially available, and then you find a pot of gold somewhere and you can buy it, then that's sometimes a way that you can try and get around it. But it's really tricky because if the goal is to take it to as many patients as possible, there's a certain level of academic rigor that has to be met that is driven by the FDA for it. But I think advocacy and coming at things together really helps.
Just out of curiosity, who's on the FDA board? Are there any pediatric oncologists on those review boards?
So the FDA section that tends to review, so there are different review groups, and they do have pediatric oncologists that are on them. Right now, there are some great ones, and what they have been doing with the current group, what they have been doing is, there's actually a call in, I think, a week and a half, is we've been having these regular meetings with them to basically bring them up to speed. And actually, most recently, it's been not just FDA, but EMA. It's like the European FDA equivalent, the European Medical Agency. And so they have heard very loud and clear us bring up these issues where if they're asking for, this level of rigor, and we're like, that's never going to happen, then they're really starting to hear it, and they're partnering with us, but it's through education and it's through partnership, right? So, I think one of the things that has really helped with form some of those relationships is the Accelerate Forum, which is a group that initially was started by Andy Pierce, although he's no longer leading it. But it basically brings in investigators, researchers, pharma, patient advocacy groups. There's always a topic. It's held twice a year. And the regulatory agencies with both the FDA and the EMA. And everyone's in the same room together. And I really think that through those sorts of forums, it's hearing your question, and it's hearing the families' voices and the parents' voices that sometimes, nobody cares to listen to my voice, right? There are a few companies where, I think they probably just hear me in their sleep and are like, whatever, just off, right? But if you're a parent and you're saying, why can't we do this, or this doesn't seem reasonable, it really matters, and it really helps through some of those advocacy programs.
I'm done. Anybody else have questions? Yeah, go ahead. One more question. I'm sorry, were you done?
Yes.
Okay. Okay.
On the trial that's coming out on COG, where some patients are just doing standard treatment and then the other ones have immunotherapy, starting in the second cycle, is there a way, just like there was when they compared chemoimmunotherapy to the 3 chemos, irinotecan, temozolomide, and whatever, is there a way, if you start seeing that 70% are responding 40%, can everybody switch over?
Yeah, so and that's why it's such a good question. That's why I was trying to point it out as I went, where clinical trials will oftentimes have interim analyses that are built into the trial. And usually the main reason is to make sure that we're not causing too much harm, right? That there's not too much toxicity. But there's always a part of it that is to look and see, like, is one group doing significantly better or significantly worse, in which case we need to not do the whole clinical trial. So that's how antibody, that first trial, 0032, that added antibody at the end of treatment, that was an interim analysis where it stopped early. And then the 3 drugs versus chemoimmunotherapy, that's why that stopped early. On AMBL 2131, there's also different interim time points where we stop, and it's not the study team, right, because we're biased, that looks, it's somebody else, the statisticians, that stop and they look at the data and they look at the safety and they look at the efficacy, and then they tell a different group of people about that data, and then a decision is made, okay, carry on, close the trial, this becomes the new standard, and so those are built into there.
Okay, so it could be sooner than 5 years that people on that trial, everyone's getting the chemoimmunotherapy up front. Is
Yeah,
that
I mean,
true?
there's
It
just
could
no way
be.
to know. So yeah, so the trial, in order to accrue, is going to take about 3 years, which is another 2 years from now. And then there's the 3-year before we know the answer. If there's an earlier stopping point, then it would back up potentially.
Okay, one more question. At St. Jude, what is y'all's current standard of treatment? Does it include the immunotherapy, or did y'all stop since that
Yeah,
was the
so
trial?
our current standard is AMBL 2131.
Okay,
Yeah.
thank you.
Yeah.
Anyone else? Oh, okay, one more.
So I have a question about the ANBL 2131. Is there anything, because I know it's randomized, but is there anything that would disqualify a patient from automatically going to arm B?
No. So it's randomized in what it is. We're trying to balance the treatment arms to make sure there aren't confounding factors. And so the reason it's randomized is we shouldn't be just selecting, and then we have to balance for the same number of patients who have MICA amplification, the same number of patients who have an ALK alteration, and then also the stage of disease. There are reasons that if a patient gets randomized to arm B, which is the chemoimmunotherapy arm, they might not receive the initial immunotherapy. So this actually just recently happened with a patient of mine who still had because of their cancer, had a lot going on in the lungs. And so there are criteria on when you should or should not give antibody. No one wants to make anyone worse. And so for that patient did not receive it in the second cycle, but they were on arm B, but then they picked up and started getting it after that.
Okay.
And so just kind of the same thing even with other chemotherapies, right? There are certain reasons that you might not give certain chemotherapies if a patient has a lot of toxicity or one of their organs isn't doing well.
Okay. Yeah. Thank you.
Great.
Okay. All right. Thank you,
Thank
Dr.
you guys so
Federico.
much. I appreciate it.
Thank you.